Okay, so sorry, this feels pretty weird. It's a bit of a shift back to oceanography, and then another shift because we're going to start talking about biogeochemistry and carbon in particular. So I'm Elisabeth Shadwick. I came from Hobart. I work at CSIRO, which is the National Science Agency in Australia. I'm also affiliated with the Australian Antarctic Program Partnership, which I think some of you are familiar with or even contributing to. Thank you very much for having me. I appreciate the opportunity to be here. I haven't taken up the theme of talking about myself and my career path. I might be persuaded to do it tomorrow, but I did want to just mention a couple of things. So I was really delighted to hear about applications of the data that's collected by the SOFS mooring, which stands for Southern Ocean Flux Station. I did want to plug, hopefully not too self-servingly, that that's part of the Southern Ocean Time Series, which is a long-term deepwater mooring program that I run out of Australia. And I did also want to plug that the data is all freely available, and the more users, the better. And we just came back a couple of weeks ago from recovering, that's it there, dirty and biofoul, the 12th SOFS mooring. So we now have a record that is a little bit patchy, but extends longer than a decade, and deployed SOFS 13. And the reason for showing this team photo, I did just want to take a little minute to pick up on the theme that Seb introduced at the beginning of his talk about the diversity of the people who are here at the summer school. And I agree that it's really wonderful to see this spread of individuals. I don't know if it comes across well in the photo in a dark room, but this is me. And you know, we're all similarly dressed in extreme high vis, which is now necessary on Australian ships. But something to point out, I don't know how many people are in this photo, you could try and count them. I'm the only woman. So I am the chief scientist on these voyages that I do every year. And I spend 3 weeks entirely surrounded by a team that looks like this. And these are the people who help on the deck. A couple of them are scientists. Many of them are part of the ship's crew. And the Australian program, I'm sorry to say, has a pretty terrible record with diversity of people who work on their ships. So it's not a complaint, but rather an open invitation to anyone, female or otherwise, who wants to talk about challenges of working at sea and the way that diversity sometimes falls apart depending on the discipline that you pursue and where you choose to do your work. So happy to discuss that at another time. For today, so I was asked to talk about the biological carbon pump, I think, and I've tried to adhere to that topic broadly. So I will talk about how biology, and that'll require a little bit of a reminder about the physics drive carbon cycling in the Southern Ocean, sorry. And if I tend to talk really fast, so if that seems like it's happening, someone could wave at me and I'll try to slow down. Partly it's nervousness, partly it's just a style thing, so apologies for people, especially those who don't have English as a first language. Oops, sorry. Then the next bit to cover, I think the pointer does work. Yeah, the next bit to cover is the biological carbon pump. So that's probably a term and hopefully a theory, if you like, that's familiar to most people. I've written pumps there because I'm going to go into some of the emerging ways that we're starting to think about this a little bit more sophisticated way, which relates to new ways of measuring the way that particulate carbon moves in the ocean. And then I'll finish off by relating the biological carbon pump, or the way that biology moves carbon in the ocean, to ARC CO2 fluxes, which hopefully will link nicely back to some of the things that we've talked about the Southern Ocean in terms of heat and carbon uptake. And I won't talk about what I'll talk about tomorrow. I can leave that till tomorrow. Okay, so increasingly, I feel that my own research, which is really focused on understanding the changes in the Southern Ocean carbon cycle, is motivated by these two great services, both the heat, which is of course not my area, and carbon uptake. And so we heard about this from Matt and Sarah and others. This is a kind of, I guess not so sophisticated cartoon, but I like the way that it shows the energy imbalance that we have. So this is of course due to anthropogenic activities. So the values that are listed are between 1971 and 2018, and then a more modern period in the parentheses there. And what you see really clearly is that the ocean has taken up about 90% of the heat that we've put in the atmosphere through the burning of fossil fuels, right? So that's not a surprise to anybody in this room, but I think it is important to remember how massive that is compared to where the heat is stored in the other reservoirs. We have a kind of similar story for carbon. It's not quite so stark, I guess, as the ocean, but this is an update from the recent, so the 2023 update to the global carbon cycle. So the Global Carbon Project is a plug to them. This is an update that happens every year. They do these budgets and update the numbers. So a couple things to point out. So the big blue arrow here, that's the fossil fuels that we're emitting to the atmosphere. And then the circle. So the size of the circle is proportional to the amount of gigatons. So I'm going to talk about the ocean today. Clearly, I hope clearly, dissolved inorganic carbon, or DIC, is by far the largest reservoir of carbon on the planet, right? So it trumps the oil and gas that we haven't yet dug up and burnt and emitted, as well as things like permafrost, soils, surface sediments, and particularly it dwarfs organic carbon. And if you can squint your eyes and see this very tiny dot, that's the marine biota, right? So by far, orders of magnitude, the biggest reservoir of carbon on the planet is in the form of dissolved inorganic carbon in the ocean. And that's important, especially because it's able to actively exchange with the atmosphere. So I know I was asked to talk about the biological carbon pump, and I will get to it. Okay, so this is a reminder that the carbon sink is, of course, controlled by physics and biology. And we've had lots of great talks about this already. So this is a figure that's familiar now. And we had lots of great introduction of the different water masses as well. The thing I want to point out here is that the ocean sink for anthropogenic carbon, and to some degree natural carbon as well, and I'll get a little bit into the differences, is controlled by the large-scale physical drivers, or the physical ocean circulation, right? So that anthropogenic CO2 comes in from the top, and the way that it gets moved around is in response to the ocean circulation. Now, natural gradients in dissolved inorganic carbon, particularly if you think about them with respect to latitude in the Southern Ocean, are also controlled by large-scale physical movements or circulation. But the biological carbon pump, and this is a really simple cartoon of it, and I'll talk about it more, control the vertical gradients in natural carbon. So that has been really important for long time periods, so glacial, interglacial variations in ocean carbon, for example. And so while these processes might not be the things you think of in the short term, I think there's an increasing recognition that there's a need to understand how carbon is moved from that big reservoir of dissolved inorganic carbon into those tiny ones of marine biota and organic carbon to fully understand the processes that allow the atmosphere and the ocean to exchange. Okay. All right, so thankfully, I don't have to really start with physics because the physics has been very well covered. But this is the cartoon that we're all kind of getting used to, where you have nutrient, and I would specify also carbon-rich deep waters that get upwelled south of the polar front. So that's that lower cell that Matt was talking about. And then some of those waters are subducted, of course, so north of the subantarctic front through the formation of mode water, and also close to the Antarctic continent. So partly that's via that dense water formation that's important for sea ice growth. And so carbon moves with this water as it does its circulation in the ocean. This is a kind of zonally average picture, the one on the left that we're all kind of used to, but also no, it's not very realistic, right? So I really like this review by Alison Gray, which came out late last year, so I'd encourage anyone to have a look at that. And the fourth dimension here, so it kind of beautifully shows, and I like the pink and blue colors, the way that the water subducts and moves forward with transport, and you've got the westerly winds, and she starts to pick up the distinctions between the different basins, and then you've got storms, small scale variability like Seb talked about, mode water, CO2 outgassing at the surface, and ingassing, and then every once in a while these little green dots that indicate biological carbon uptake. And the fourth dimension here is, of course, time. So I think it's important, and like we've heard already from other people, to remember that the timescales that we're dealing with vary across a really wide range. So day-night variations in heat fluxes, or if you like, in oxygen from phytoplankton converting their photosynthesis to respiration. Then you have the passage of storms, which influences mixing and the properties of the water, like carbon, that are influenced by storms as well. Then you have things like this seasonal ramping up of phytoplankton productivity, so development and cessation of blooms that can be over kind of months or seasons. And then you have these larger scale climate drivers, things like the southern annular mode that Matt introduced, that are operating on more decadal timescales. So people have mentioned this already, but I do think it's important to reiterate that if we want to understand the processes that are driving these static pictures that we like to build, we really need observations that go from short-term hours to maybe days, to weeks, to months, seasons, and also over decades. So observing these things is a challenge, but it requires commitment to do so. Okay, so still physics, but now with some biogeochemistry. This is another paper that I really like, and I would encourage people to read it if they haven't done so. So this was about really, I won't say discovering, that's probably a bit too strong, but really identifying subantarctic mode water as an important supply of nutrients and also oxygen to the low latitude Southern Ocean. So the idea here, without going into too much detail in the figure, is that biological processes, of course, strip nutrients out of the surface waters, right? Phytoplankton grows just like plants on land. They require CO2, of course, but also nutrients to grow their soft tissues. And then some of that stuff sinks or gets eaten, and the carbon and nutrients are exported into the ocean interior. I'll come back to it later. But if there was no return path for these nutrients, then biology would very shortly strip the surface ocean of nutrients, and that would be that, right? So there have to be return paths for these things. And this paper in particular identified this subantarctic mode water, which is important for lots of reasons, but here's another one, as a return flow to provide nutrients outside of the Southern Ocean, as well as locally. Oops, sorry. Okay, and so this is a more modern version of such a cartoon. So this is a relatively recent review that I was involved in that was led by Philip Boyd about the role of biota, so that includes kind of all organisms in the Southern Ocean carbon cycle. And so here, what's being shown is what we now refer to as a biogeochemical divide. So I hope you can see that up here, which is kind of at the polar frontal zone. And that's associated with high organic carbon export, so particulate organic carbon that rains out of the surface waters. And silica in particular is used by diatoms. So south of this biogeochemical divide, we have these highly silicified diatoms that are typically a good ballasting material, so they get heavy and clump together and sink, so they're considered to be efficient exporters of organic carbon. And this happens kind of at the southern edge of the ACC. You also, of course, have CO2 outgassing, right? So I mentioned that upwelled nutrients also upwells carbon. So you have outgassing of natural CO2 associated with these upwelling events. Some of this is offset by NPP, sorry about the acronyms, that's net primary productivity for those who don't know, and the subsequent export of particulate organic carbon. There's also an influence of calcification, that's particulate inorganic carbon, so that's these other guys that are shown as these spherical things, coccolithophores, who make structures out of calcium carbonate. And that actually produces CO2, and that's part of the carbonate pump, or the carbonate compensation pump, if you like. And I'm not going to go into that in a lot of detail today, but I would be delighted to talk about it with anybody who's interested outside of this. So that's the, again, zonally average kind of static picture that we have about these processes. So that's physics driving biogeochemistry to some degree. And now we'll move on. Oh, yeah, I did want to say this because I think it's important. So this is the silica trap. So because these diatoms use lots of silica and then they sink out of the surface layer, silica does not have a chemical dissolution the way that inorganic, particulate inorganic carbon does in the atmosphere, or in the ocean, sorry. So when those diatoms sink, they of course are eaten up, and some of them are acted upon by microbial processes that want to return silica to the water column, but there's no chemical dissolution for biogenic silica in the ocean, so there's no lysocline, if you're familiar with that term. So there's no point at which the mineral just begins to dissolve. It requires a biological process to return that inorganic silica back to the water. So what happens is a kind of silica trap where a lot of it gets stuck there and a lot of it accumulates in the sediment. And indeed, more than 2/3 of the total ocean silica burial occurs just under this band in the ACC. So I think that's why Australians like opals, because they find lots of opals in Australia, and that's a result of these marine organisms that sink and stay well preserved. Okay. Sorry, I'm having issues with the clicker. Okay, so I just briefly, I was happy to see that Matt mentioned this, and this is another example of a physical driver of ocean biochemistry, biogeochemistry, but operating on relatively longer timescales, right? So this is just another cartoon of the same picture that we're all very used to by now. And the bullet points there just describe what happens in the positive phase of the southern annular mode. So this is a dominant climate mode, as Matt explained to us. It also has impacts on the biology, or biogeochemistry, if you like that term, not just the ocean circulation. So just really briefly, so we're talking about a positive SAM. We have westerly winds in the Antarctic zone, so that's this zone pretty far south here. And in the polar frontal zone, that's defined as this one between these two fronts. And you get an increased equator transport. So that's that Ekman transport that's moving stuff north. So what that also has associated with it is increased nutrients. So forget about iron for a second. Increased nutrient supply, because you have pushed water this way, so you need to backfill it with something. So you upwell these waters that are nutrient rich, and that allows an enhancement of net primary productivity. The opposite is true north of the subantarctic front, or up in the subantarctic zone, which is where I actually do most of my own research these days. So you have a deeper mixed layer here, which is likely to lead to a reduction in the supply of nutrients coming up the previous winter, for example. And also the light environment changes. So we're likely to have decreased net primary productivity in those times. So you have this contrasting effect on the amount of organic carbon potentially available for export depending on the phase of the southern annular mode. So this is intended just to reinforce the idea that physics does, yes, drive biogeochemistry. Physics, yes, does set the large-scale concentrations of the things that we care about, but the biological processes, we do need to understand them in order to really get at the full understanding of the system. Okay, so why is the Southern Ocean special? For lots of reasons, of course, and we've heard about lots of them already. This is another old paper that I like and I would encourage people to read. And I love the quote at the bottom here. So I'll just explain the figure. So the figure is showing export production. So that's grams carbon per square meter. And in this instance, it's the amount of particulate organic carbon that reaches below a depth of 500 meters in the ocean. And so Falkowski says the export of organic material from the euphotic zone to the ocean interior, followed by biological oxidation of organic carbon, leads to an inverse concentration gradient in organic carbon. So then, so just to sum it up, that is the concentration of inorganic carbon below the upper 500 meters of the ocean is greater than that at the RC entries, right? And so probably anyone who's taken a chemical oceanography class feels like they know that. You get this subsurface maxima in dissolved inorganic carbon that results from the remineralization of organic material. And that means that at the surface of the ocean, because the phytoplankton has used that inorganic carbon to build their bodies and do their thing, that puts the ocean at a lower concentration than the atmosphere, and that allows CO2 to come in from the surface. So of course it is more complicated than that, but I do love the simplicity of this explanation. We heard a little bit about this, I'm sorry, I can't remember from who, but the Southern Ocean is also special because it is by far the largest high nutrient, low chlorophyll region in the world ocean. So this is just a NASA image of chlorophyll. And I have to admit that looking at this, you wouldn't necessarily conclude that the Southern Ocean was the largest high nutrient, low chlorophyll region because you still see plenty of chlorophyll. And particularly you see hotspots of productivity on the Antarctic shelves, which I won't talk about today. But let's say we believe that it's a high nutrient, low chlorophyll, and you can believe me when I tell you that 50% of the nitrate and phosphate go unused in the Southern Ocean, and that's because, or at least partly because, the phytoplankton there are limited by an important micronutrient, which is dissolved iron. And Al, who is going to give his lecture remotely, is undoubtedly going to talk a lot more about this. But I wanted to say this because of something that Matt brought up, that if we assume that these high nutrient, low chlorophyll conditions are because of iron limitation, then you can go on to think, and I know there's at least one paleo person who's in the audience, you can go on to think that if you had an enhanced delivery of iron during glacial periods, and there is some proxy evidence for that to the Southern Ocean, you would stimulate primary production, right? They have underutilized nutrients, they require iron, you give them some iron, they do more primary productivity, and they could then, in theory, draw CO2 down from the atmosphere. So that would lead to an enhanced export of organic carbon, so you get more organic carbon into the ocean, that can allow more CO2 to be in the ocean, and less CO2 in the atmosphere, which that's colder, right? So that's what we think happens in a glacial period. So I know that Matt said something about CO2 solubility being the major player, but I do think that the broad consensus in the community is that you need to do more than just change the temperature and deal with CO2 solubility. You also need to have enhanced biological productivity to make the full change in CO2 that we observe in the ice core records. Okay, so the biological carbon pump is important. Here is a cartoon that I would have seen when I went to graduate school. So it's pretty simple representation, something that you might have learned about if you ever used a NPZD, so nutrients, phytoplankton, zooplankton, detritus, or sedimentation, if you like, type representation in a model. So this is a paper by Sally Chisholm in, I think it's Nature. Yeah, so the hilarious thing is when I went back to get this figure, I realized that this paper is actually part of an introduction to an issue that was all about the potential evils of iron fertilization. And I'm going to talk about that tomorrow. So that is something that has come back around quite swiftly, probably. And this figure is shown all the time as an example of the biological carbon pump. I just thought it was interesting to note the context for which it was originally drawn. So this is a really simplistic representation. So you have sunlight, which is the energy source for photosynthesis that's done by large phytoplankton and small phytoplankton at the surface of the ocean. And by doing this, they allow CO2 to come in because they convert the inorganic carbon to organic carbon. Some of them sink and are remineralized or respired by bacteria. You get this smaller loop here between small phytoplankton, bacteria, and microzooplankton, so that is now referred to as the microbial loop. You got a bit of interaction here with zooplankton who eat the phytoplankton, mostly, but not always the large stuff. And then fish who eat zooplankton. Fish, of course, can also eat phytoplankton. I don't know why there's no arrow in this one, just because it was supposed to be simple, I guess. And then whatever escapes either consumption by somebody else or respiration by bacteria is free to be exported out of the surface ocean into the deep ocean. So perplexingly, also, there's nothing between the surface and deep ocean in this depiction. And there are some critters who live down here who would then derive energy from the delivery of organic material into the deep sea, and then very little actually makes it all the way to the sea floor. So most of it is turned around in the upper ocean. Some of it escapes down to here. The idea being that if it escapes this surface ocean, and depending on where you are, that can be very deep, right? So in the subantarctic Southern Ocean, it needs to get down to more than 500 and probably more than 1,000 meters before it's really considered to be sequestered from remineralization back to inorganic carbon or CO2, and then subject potentially to re-exchange with the atmosphere, right? In order for this to really be the way that we get atmospheric CO2 into the deep ocean, either the organic material itself or the subsequent products of that organic material upon respiration need to be very deep in the ocean. So this is one of the issues that would come with something like iron fertilization, where just enhancing primary productivity at the surface is really not enough to get additional carbon sequestration in the deep sea. Okay, and then this little extra on the side here is a really overly simplistic, of course, one-dimensional view of, let's say, the solubility pump or the way that carbon is moved by physics. So won't talk about that anymore for now. I did want to spend a little bit of time talking about the ways in which we can measure these things in the real ocean. So has anyone used a sediment trap? Woo! Okay, yay, all right, a couple of people. Martin, really, okay. So, yep, so these are, McLean is just one manufacturer of sediment traps. There are others. These ones probably are the most recognizable because they're big yellow funnels, so it's, kind of taller than I am. It has a rotating carousel down here at the bottom that can have something like, between a dozen and 21 cups. And the funnel is not totally open at the top. The funnel has a kind of baffle, we call it. It's a kind of coarse mesh that's supposed to prevent critters from swimming into the funnel, although I will say that we catch fish more regularly than we plan to. But it's supposed to limit the delivery of material in through the top of the funnel to the small stuff, right? So the marine snow. So the idea is not to capture large organisms, but what's left over after those turnarounds have happened. So the material comes through the funnel, and there's another little filter at the bottom here. That's the motor. Or maybe that's the control and that's motor. Anyway, this thing rotates so that for about a fortnight, if you want a full year of observations, you have one open cup under the funnel. And before you put the thing out for deployment, you fill the cups with a really highly concentrated brine solution that also has some kind of chemical preservative. We use mercuric chloride, which means that whatever falls into that cup is instantly killed in case it's still alive, but it also means that microbial degradation of the material can't continue while it sits in that cup waiting for it to be picked up, right? So you get to assume that whatever material has fallen in there and you have caught back when you recover the trap is representative of the material that was falling into the cup when the thing was out in the ocean. And then the brine solution is just so you might ask, well, if the thing is open, what happens if seawater flushes in? So you make it a really salty brine in the cup so that the material stays in there, so it can't get flushed out by the surrounding seawater. So that's a kind of old school conventional way of catching, if you like, particulate organic carbon, in the ocean. Increasingly, people are using BGC Argo floats. I don't exactly know what to call this one. I think it's called a profiling sediment trap. And so the buoyancy engine is something that looks very similar to, and I believe is very similar to, an Argo float. And then you have these collection devices around it. And then the last one is similar to the photo that Seb showed of a very sad, tired glider that was just recovered in April after having been 5 months in the Southern Ocean. And this was after a first pass of cleaning, so it really was covered with gooseneck barnacles and very biofoulled. But these BGC Argo, this one has collection cups, and this glider, so they now carry so-called bio-optical instrumentation. So particulate organic carbon is estimated from a backscatter sensor, and there's quite a lot that goes into getting from backscatter to particulate organic carbon. But assuming that you might believe all those assumptions, that potentially gives you an estimate of particles, and to some degree particles that can be distinguished in terms of their size. And they can also carry other sensors, like I've heard Seb mention an EcoPuck, which also has chlorophyll fluorescence. And that one too, if you wave your hands around, you might believe that that one gives you a proxy for biomass. And so you get chlorophyll, which maybe gives you a proxy for biomass, and you get backscatter, which maybe gives you a proxy for POC. And if you put those two things together, you can maybe convince yourself that you're doing the same type of measurement as physically collecting the particles as they sink in the ocean. Okay. So this is about, what is it, from 2007. Okay, so almost 20 years old now. And it was just intended, I think, to give an overview of the processes that were responsible for controlling the export of biogenic particles, so POC basically, but it could also be biogenic silica, like the diatoms that I talked about. And so it's, we have particle production in the euphotic zone. There's physical aggregation, so they clump together. They're detrained from the mixed layer. This is something I'll come back to. Some of them settle, and organic matter is remineralized. Some of it makes it past the lysocline. That's that chemical threshold there where particulate inorganic carbon would start to dissolve. And what I did want to focus on a little bit is the horizons and timescales, right? So within the euphotic zone, these particles could maybe last a season. Once you get outside of that, below the depth of the summer mixed layer, they might hang around for about a year. It's really once you have gotten below the winter mixed layer, which of course varies depending on where you are, that you have storage of this material at decadal, I would say that's probably longer for mode waters, or centennial or even millennial timescales, depending on how deeply you manage to get that material before it is remineralized back to CO2. Okay, so this is just one example of a relatively recent paper by a colleague of mine, Catherine, where she looked at a long time series of POC export using a McLean sediment trap from time series observations. So this is the Southern Ocean time series. It's where we have the RC Flex mooring. It's in the subantarctic zone, so southwest of Tasmania. And this is one of the longest Southern Ocean time series that I'm aware of. So we just deployed the 26th SAS sediment trap mooring. So this has a surface float that's about 800 meters below the surface, then paired sediment traps at 1,000, 2,000, and 3,800 meters. And so combining all of this data collected over these many years and doing a kind of stacked seasonal climatology, you get something like this, where the flexes, the particulate organic, so milligrams carbon per square meter a day, by the blue bars and by month. And then this net primary production, oh, sorry, it's not coming out very well, but there's a green one, which is based on this type of satellite remote sensing algorithm, and the orange one, which I think is a little bit clearer, which is based on this one. So just very briefly, what you see is very low delivery of material in winter, and then when productivity ramps up at the surface, you get enhanced delivery of particulates to depth. There's a surprisingly small lag between the onset of productivity at the surface and the delivery of particles to 1,000 meters, and actually no lag between the delivery to 1,000 meters and 2,000 meters, which we can talk about offline if you want. And then the productive season kind of persists till about March, maybe sometimes April, and then the whole thing starts up again. Just something that I'll come back to is that it doesn't ever go to zero. So there is this kind of continuous rain of material out of the upper ocean, even though we know that it's not an active season of productivity. So that's kind of an open question that's being looked at more and more clearly now. Okay, so this is about the biological carbon pump, which is now considered to be more than just the gravitational settling of large particles and really contributions from, I don't know, Philip says multifaceted, but these various particle pumps that contribute to the whole delivery of organic material to the interior of the ocean. So this is a cartoon that's intended to show, so this is the kind of old school way of doing things. You go out with a ship, you might deploy a sediment trap. Biology is patchy in the ocean. You try to measure it the best you can. There's this idea of a statistical funnel, which kind of tells you that if I put a sediment trap down here, it's the material that I receive is representative of a funnel of of material. So the deeper you go in the ocean, the broader the area at the surface that you're assuming you're representing by the material that you receive. And then these things are extrapolated using, that's kind of 1D view, they're extrapolated using satellites maybe, or models possibly. This is the kind of newer view of these things. So it's not all about gravitational settling. So we don't say biological carbon pump anymore, but tend to say biological gravitational pump. And then there are these other ones: eddy subduction pump, mixed layer pump, mesopelagic migrant pump, and seasonal lipid pump. And they all operate kind of differently, but ultimately they all contribute to the delivery of material, deeper than the permanent picnic line. And then there's just a last little thing, which is about being able to detect the contributions from these other pumps requires different technology or different instrumentation than the conventional sediment traps that we have used over these, last 50 years or so. Okay. So this is now an ocean of many pumps. So not just the biological carbon pump. And you see that this is a somewhat more complex version of the first figure that I showed you, where there are a lot of different processes that act on the material as it makes its way within the euphotic zone and then out of it. And so I would just like to point out, so the particulate organic carbon in the surface ocean provides food or energy to the mesopelagic ocean. So that may not be where the majority of fisheries, we talk about fish that we like to eat, live, but there is a significant biomass in the mesopelagic ocean. And it's only the fraction that escapes remineralization in the open ocean that can possibly be a food source to the mesopelagic, right? And so part of this development of this many pumps which contribute is motivated by a need to reconcile what we think is the demand for energy, or the demand for oxygen, if you like, in the mesopelagic with the delivery of particulate organic carbon. So our estimates of particulate organic carbon delivery from mostly sediment traps and other ideas about gravitational settling were not sufficient to meet the demand that would be necessary for what we think is the biomass in the mesopelagic ocean. So it seems that invoking ideas about these other pumps, whether they be the mixing, the eddies, or the mesopelagic migrating pump, it's necessary to close that loop. So if we only think about gravitational settling, there's not enough food for the stuff that we know lives in the ocean. Okay, so this is just, this is the first one, the mixed layer pump. Sorry if it's hard to see. Schematically, this is what happens. So you have, this is phytoplankton, say, in the surface of the ocean, and this is the mixed layer, which changes over a season. So when the mixed layer is shallow, you have lots of light, maybe this is spring or autumn, you've got stuff growing, and then the mixed layer suddenly deepens. When it deepens, it redistributes this material over a much deeper fraction of the upper ocean. And then when it restratifies, some of these particles are left behind. So that's a kind of physical way of getting particles into the deep ocean that doesn't require them being heavy and sinking. So this is thought to mostly act on small phytoplankton, not the bigger groups. And the next one here is about the eddy subduction pump. So we heard about eddies yesterday, where you have dense and less dense water. And the idea here is that eddies promote subduction. So you have kind of short or potentially long term. They can be small, they can be large, but you have a mechanism for getting water from the surface of the ocean into the interior. And you can imagine that it's going to carry its properties, which include particulates, if they're there, into the ocean interior with it. So that one's the eddy subduction pump. And this is a recent paper by Léo Lecour, when he looked at BGC Argo float observations over several years, so from late 2016 to 2019, so over several productive seasons, and was able to quantify the different mechanisms for particulate organic carbon to get into the ocean. So the light blue, these colors will be terrible, sorry, but let's say the first triangle is the onset of the spring bloom. This is meant to be the peak of the spring bloom, and that is meant to be when it kind of terminates. And so the idea here is that they observed 1, 2, 3 of these productive season events, quite different, right? The first one has a very obvious ramping up of POC and chlorophyll, and the others are relatively flatter. I guess this one comes a little bit late. And this panel in the middle is the POC over here, that is the blue bars, the solid blue bars, that is delivered by the gravitational pump. So notice this is a log scale and also the axes, and this is an attempt to estimate how much is delivered by the mixed layer pump and the eddy subduction pump. And so you can see that the contribution is not zero. It's relatively smaller than the gravitational pump, but it's probably still important if you consider the whole year. You see that the mixed layer pump was pretty important in this first blue, maybe a little bit here, but less so over here. And another takeaway is that this one seems to operate outside of the productive season. Same with the eddies. So the eddy events tend to be quite sporadic. I guess you already know that if you study eddies. And so these subduction events can have a big influence on delivery of particulate matter to the ocean interior, but they're not very easy to predict, and they don't occur that frequently. All right, so this is, I think, important because the so-called mixed layer pump or seasonal mixed layer pump is really going to influence the delivery of organic carbon into the ocean interior in regions where we have substantial changes in mixed layer depth. And so we know that the Southern Ocean is one of those places where you can have very rapid changes in mixed layer depth, and seasonally we go from waters that are quite stratified to waters that are mixed very deeply. And so this paper concluded that in regions of deep mixing, and I like this figure at the bottom in particular because it's back to our old sub-Antarctic zone here, where we know the mixing is important for mode water formation and other reasons, that they possibly account for 20, so that's almost a quarter, right? So almost a quarter of the POC supplied by fast sinking particles. It could be that this is, again, the export production, okay? So this is, I think this is down to depth below 500 meters. So particulate organic carbon, so you get the big splashes of it in red and yellow, and they line up really nicely with places where there are deep mixed layers, particularly in winter, of course. So this is probably an underappreciated mechanism for getting inorganic carbon into the ocean interior. And this is... sorry, this one. This one, that'll be a model. Yeah. Well, model. It could possibly be based on some satellite information, but yeah, that'll be a model. And I suspect this is a reanalysis product. Yeah, I'd have to look to be sure. Yeah. This is definitely a model, and so this goes back to Seb. You had a whole thing about mesoscale, importance of mesoscale and eddies, and I like this one because this is again the region that I work in, so it's south of Australia. So these are snapshots again from a model. So this is export production, so material that makes its way past 500 meters in the ocean, and this is the mixed layer depth. And this is a change from some day in October, the 1st of October, to the 1st of November. So within a month, you have mixed layers that are mixed layer is over here. So you go from very deep mixing, which is what we're used to. I'm getting it backwards, I think. Majority of large, okay, sorry. I'm getting confused. So these are large signals of export production coming in November that are associated with an onset of stratification. So when you have very deep mixed layers, you don't get material sinking out of the ocean interior, but you can rapidly stratify, and that can have a pretty fast response by the ocean of dumping this particulate to depth. And so this is probably spring. I mean, this is well, it is spring, but this would precede the bulk of the productivity that we would expect to happen over the summer. So you can get short but really important events that deliver a lot of material into the ocean interior. Okay, so how am I doing? Yeah, this one, a bit about zooplankton. So I did say that I would talk about zooplankton. So this one is a little harder for me to understand because I don't really work on zooplankton, but let's have a go. So this is about a so-called seasonal lipid pump. So the idea here, this is from the North Atlantic, but these are copepods, but I think it would equally apply to krill in the Southern Ocean ecosystem. So you have copepods growing at the surface, and they produce lipids, which is important for them because then they do this deep migration and they spend some fraction of the year kind of hibernating at depth. And the idea here is that they use nutrients while they're accumulating lipids, but those tend to stay in the surface. So they eat things and they produce lipid in their own body, and then they do some respiration, and so they don't actually take much carbon and nitrogen with them. And when they get down here, they then use their lipid reserve to stay alive in winter. And this releases carbon, but apparently not nitrogen. So there's a decoupling between the delivery of carbon and nitrogen to the deep ocean, which has implications for leaving the nutrients or returning the nutrients to the surface where they can be used by primary producers, but also having carbon exported to depth while these guys are down there during their hibernation period. And this is a bit less relevant, but I did want to show it just briefly because I think it's really cool. So this is work that was done recently by a former student who's now a postdoc at NIWA, Svenja, where she did some experiments on how influential the sinking of dead zooplankton are to the delivery of particulate organic carbon in the ocean interior. So these were experiments that were done at not simulations, but tank experiments that were done from collected zooplankton at 1,000, 2,000, and 3,800 meters to line up with the locations of the sediment traps. And what she found was that despite the fact that they have really high rates of carbon turnover, the delivery of these carcasses, so the delivery of these dead zooplankton, actually make a contribution that is significant to the organic carbon flux at depth because they do sink rapidly. So they're being turned around by microbes as they sink, and that is a potentially not negligible fraction of the carbon pumping in this region, subantarctic. Okay, so a bit more about zooplankton. I couldn't help myself. So this is a recent paper by a former postdoc, Tyler Rohr, and I love this figure. It's too complicated to explain in quick, but just looking at it. So what he did was he evaluated 11 CMIP models, biogeochemistry models, and what you see in here are the different ways in which they parameterize things that are important to zooplankton. And what he found was, quite spectacularly, I think, that the largest source of intermodal uncertainty when you look at the carbon cycle of these various models is about how they parameterize zooplankton grazing. So I can't remember who said it. I think it was you, Dirk, that we don't always need to make things more complex, right? So this was not about adding different classes of zooplankton. This was not about going from, one class of phytoplankton to 12 classes of phytoplankton, or one class of zooplankton to 3 classes of zooplankton. This was really about identifying that you could reduce the spread in how the models represented the carbon cycle if you got really specific about how they parameterize zooplankton grazing, so the way that zooplankton eat phytoplankton in the models. And so this is for the Southern Ocean in particular, where the green is net primary productivity, the blue is export production, and gross secondary production, so production by higher trophic levels, in this case zooplankton. And this is gigatons carbon per year, and this is what happens if you increase grazing rates. So the conclusion here is that all of these things, so net primary production, gross secondary production, and export production, all super important for moving carbon from the surface into the ocean interior. They all collapse at a really narrow range of grazing parameterization. So getting this one thing right in the models would really help us to have a more consistent understanding of how carbon moves from the surface into the ocean interior. So getting something as simple as how quickly do phytoplankton specific loss rates, so how quickly do the zooplankton eat the phytoplankton, makes a massive difference in the agreement of these models in terms of how they move carbon in the Southern Ocean. All right, getting there. So this is, I'm getting close to the end here, because I know I'm standing between you and lunch. So I had said that I would talk about linking the Southern Ocean biological carbon pump or biological carbon pumps to ARC CO2 fluxes. So this is a recent paper that makes use of this new array of BGC Argo floats, which Sarah talked about, and I'd be happy to talk about more. And specifically wanted to look at how biogenic carbon pools, so particulate organic carbon, helps to maintain the status of the Southern Ocean as a carbon sink, so talking about atmospheric carbon. And he used 11 observations of the seasonal cycle, so this is 63 independent floats that did, 11, that did 100 observations of the seasonal cycle. So just really quickly, the BGC Argo floats, they do not have dissolved inorganic carbon or CO2 partial pressure sensors on them. What they do have is pH, and we can talk about the reliability of those sensors, but let's say you believe the pH. You can calculate dissolved inorganic carbon as a function of pH. Similarly, they don't measure alkalinity directly, but you can calculate alkalinity with relatively small uncertainty from temperature, salinity, and oxygen, all sensors that the floats do have. And nitrate is measured directly, and we talk about that one too. And particulate organic carbon can be estimated from backscatter. So assuming that you're willing to go with these assumptions, you can do some pretty spectacular things with this array of floats. And in this case, what they did was, bear with me because this figure is complicated, but in this case, what they did was they came up with estimates for each of these zones, so the seasonal ice zone, polar Antarctic, subantarctic, subtropic, so all these donuts around the Antarctic. They came up with estimates of export from organic carbon, from particulate organic carbon, from dissolved organic carbon, and from particulate inorganic carbon. And they related that to what it would look like if the Southern Ocean was abiotic, if there was no production of particulate organic carbon. So the observed ARC fluxes are this much, 0.4. This is the physical movement of carbon around the place. This is organic carbon, and this is inorganic carbon, which opposes the organic carbon production. But the very, I think, impressive conclusion from this work is that if you don't have organic carbon production in the Southern Ocean, it is not a sink for atmospheric CO2. So even though at the beginning I said what's important is the way that the physical circulation moves the carbon around, and the biological carbon pump is really only there to set these gradients that maybe matter over long time scales, that's probably not true. If you would shut off the production of organic carbon, not organic matter, but of particulate and dissolved organic carbon in the Southern Ocean, it would not provide us this service of absorbing CO2 from the atmosphere, which is pretty important, right? So while there are a lot of assumptions that go into this, and the numbers do not really agree with other assessments that have been made for the status of the like for the amount of CO2 flex that goes into the Southern Ocean, I do think that it does reinforce very clearly the need to consider both the production of organic carbon and the way that it gets into the ocean interior to reconcile the Southern Ocean carbon budget, which, as we know, is very important to understanding the global climate. Okay. Yes. Phytoplankton. Yep. Is that what you're asking? Yes. Yeah. So particulate organic carbon, probably the best way to think about it is phytoplankton. Algae, it's not quite algae, but so the little microscopic plants that grow at the surface of the ocean, their bodies, so maybe you've heard the term soft tissue pump? No? Okay. So they convert inorganic carbon into particulate organic carbon, the little plants that you hold in your hands. Yeah. Yeah. Does that help? They convert the inorganic carbon to organic carbon, yeah. So the organic carbon is themselves, right? I mean, the way that you are a very large particulate organic carbon. Yeah. Okay, so just briefly, and I know that Jens will touch more on this, but I do like this figure, even though it's a little bit outdated now. So we all know that we have an anthropogenic perturbation to almost everything these days, but certainly we have an anthropogenic perturbation to the Southern Ocean carbon cycle. And so this is a relatively old paper now by Nikki Gruber, but what it shows, the red colors, is the anthropogenic component of the dissolved inorganic carbon. So this is based on not something like a BGC Argo float, and I don't remember who asked the question the other day, but no, it's not possible to use the BGC Argo array to put exact numbers on the amount of anthropogenic carbon that's in the ocean. But one way we can do it is by going out and repeating hydrographic sections. So Yulia and others have recently returned from one such voyage. So we make measurements of dissolved inorganic carbon in the ocean, and we do this over large spatial scales, and we go back 10 years later and do the same thing. And the difference then, if we do some assumptions about what should and should not change, can be attributed to the extra inorganic carbon that's in the ocean that we know has come in from fossil fuels. So there are a few other ways of detecting that through isotopes and other complicated signals, but this one, sections taken 10 years apart, for example, show you, and this is a very clear indication that the anthropogenic carbon that has come in through the surface of the ocean has penetrated to depth in all of the basins, right? So you've got the Pacific, Indian, Atlantic oceans around the Southern Ocean in the middle, and you can see that this signal now has penetrated into the open ocean. So it definitely came in from the top, and the way that it moves around is due to the circulation. Yes, Andrew? I would say that the fundamental limitation is that they don't measure inorganic carbon. They measure pH. And so while I do think that most of the changes in pH that you showed over that relatively shorter record of the BGC Argo array are likely to be attributable to inorganic carbon changing by anthropogenic additions, I guess I should say these are small signals, right? So if you look at this scale, it goes up to a maximum of 15, and there's really only just a little bit that's 15. And that's in a background of DIC in the ocean, which is on average 2,100, 2,200 micromoles per kilogram. So the measurements that are required need to be extremely sophisticated in order to pick up those small signals, and that's why it takes so long to be able to conclusively observe them. And that's why it's really important to keep doing hydrographic sections, even though they're arduous and long and expensive. Yeah. Okay, so this is from Judith, who's not coming, but again, I think Jens will probably talk about it, and I appreciate that this is a really busy figure, so please just trust me and only look at the red bars here. So this is the whole of the Southern Ocean, and this is a new estimate based on observations collected and biogeochemical models run between 1985 and 2018. And what they have concluded is for the whole Southern Ocean, it's then separated into basins, that over this period, the Southern Ocean absorbed 0.8 petagrams or gigatons, if you like that, grams of carbon per year. And this figure is another old one, but I like it, from the IPCC fourth assessment report, where it shows very starkly the colors, right? So anthropogenic carbon inventory, so that's has been integrated over the water column. And you see, and probably you already know, that the big places for accumulation are the North Atlantic, but after the North Atlantic, the next most important region for the accumulation of anthropogenic CO2 is the subantarctic Southern Ocean, right? So yet another reason to care about the subantarctic Southern Ocean, because we know we have an accumulation of anthropogenic CO2 there. And I will talk tomorrow about the consequences for this, but I know that it's almost lunchtime, but I did want to go through this one because I think it's important, and I spent a lot of time with my students talking about this. So Southern Ocean heat and carbon uptake, both super critical, both really our human imprint on the global climate. But I do think it's important to just emphasize a little bit that the ocean uptake of CO2 differs from the ocean uptake of heat in a few ways that I think are important, and I think it would be great if everyone went off after this summer school with the same understanding of these things. So the first one is that the buffer capacity of seawater is a non-linear function of dissolved inorganic carbon. So happy to talk about that at length offline, but what that means in practice is that the ocean uptake of anthropogenic CO2 will saturate with increasing emissions. So it's not just something that you can keep pushing in from the top. At some point, it will slow and saturate. So that's important. And Matt, you did say something in your lecture that made me think that maybe I'm not quite right about what comes after the semicolon with this. But my understanding is that as the atmosphere continues to warm, the ocean will continue to take up most of that heat without saturation. So that's quite different from the story for CO2. We can talk about it later if I'm wrong, but please tell me if I'm wrong. And the next one, which I feel always a little bit surprised, but it's good for to explain these things, is that vertical gradients in temperature and dissolved inorganic carbon are not at all the same, right? So this is not the best image to show this, but it's the one that I could find quickly from an old paper where we did a section, the same one that was just repeated actually. And here you have a section of DIC, so south is over here and north is over here, and you very clearly see that the jet color bar that we're not supposed to use anymore, but that the high concentrations are at the bottom. So that's not the same as for heat, right? So what that means is that the overturning circulation tends to enhance heat uptake because it brings cold water up and you can get more heat in. But what the overturning circulation also does is bring waters that are already elevated in natural CO2 close to the surface. And that is more likely to enhance outgassing of natural CO2. So that's another kind of opposite. And then the last one is that the Southern Ocean, which is shown here in this review figure from Nikki Gruber and others, is that the Southern Ocean can be a source or a sink of natural carbon, depending on the balance between physics and biology, which I've explained a little bit, but I'm sure you'll hear more about that from Jens. But it's everywhere a sink for anthropogenic CO2, because that's that pervasive signal that's coming in from the top, while in the background of that, you have this complex interplay between physics and biology, which means that some areas will outgas, while others will do uptake, which is, again, not the situation for heat. So I think these things are important to go forward with, and happy to talk more about it. Okay, so this is the almost last one. So this is from, yes? Yes. Yes, the Revel factor, yes. Yeah, so not about solubility. No, it's really about the chemistry. Yeah, and so yes, about alkalinity or Revel factor, if you like that term, but basically the more CO2 you stuff in, the less capacity you have for more CO2 to go in. Yeah. Unless you change the alkalinity, which we're not doing with anthropogenic CO2, but yes, that's it. Okay, so this is from a recent IPCC assessment, and so this is just the usual depressing outlook for the future, where if we follow the 8.5, we don't call them that anymore, scenario, we get expected temperature increases of up to 3 degrees, say, and by 2100, and this is surface ocean pH, which is directly linked to the uptake of anthropogenic CO2, and I will talk about that tomorrow. So I did just want to emphasize and potentially lead into what Sarah will show after lunch as well, that the change in heat and the change in pH, these have compounding influences on the biogeochemistry and also just the biology, right? So this shows high confidence, medium confidence, low confidence, and then positive, mixed, negative. So hopefully you see ocean warming, right? So these are all negative, right? So it's likely to have negative impacts on all of these things: Antarctic krill, cold fish, benthic communities. I'll skip that one. Acidification, so not good for lots of organisms. I'll talk more about this tomorrow. Changes in sea ice, also probably challenging for some of the biota in the Southern Ocean. And then we have these things that have relatively lower confidence but might involve some positive impacts for the Southern Ocean ecosystem in particular. So that's a bit of a teaser for what I'll talk about tomorrow. And then this one, I sort of can't help myself, captive audience. So this one is about what do we need to do, right? So this is from the AR6 synthesis report, limiting warming to 1.5 or 2, and I think everyone probably agrees 1.5 is already in the rearview mirror. Involves rapid, deep, and in most cases immediate, and immediate was yesterday, greenhouse gas emission reductions, right? So this is the gigatons of CO2 emissions, and we've got time here from the year 2000 to the year 2100. And so the black is the past emissions, so this is the known emissions, right? Measurements that show what we've done between the year 2000 and the year 2015. And the red line is the one I'd like you to pay attention to. This is the trajectory that we're on with the current policies that are implemented by the various governments from the countries which we all come. And so this is not going to get us anywhere close to limiting warming to 1.5 or even 2 degrees C. And I think it is good, I mean, I know I'm preaching to the choir, but it is good to reflect on the fact that getting on to even the 2 degrees warming requires immediate decarbonization, and we're not close to doing that, right? And so these are nice pictures of the charismatic fauna and flora that you find in the Southern Ocean, and you might have squishy feelings about these things, but I do think that it's important for us as a community to remember that, yes, we want to do Southern Ocean research, and the pursuit of knowledge is really important and exciting as scientists, but it's also really important that we learn how to get the message out that this is, we're not going to get here, right? We're not going to get here on the trajectory that we're on. So I know that that's not the focus of the summer school, but I do think it would be good to have some side conversations about how we do better to convince people who get to make decisions that this is something that needs urgent action, because it needed urgent action a couple of decades ago, and that message didn't make it to the right people. That was depressing, sorry, but I'm going to finish there.